On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Creoptix AG, I'd like to welcome you to get ahead in the race with high-quality peptide discovery and binding kinetics for therapeutic and diagnostics development. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenters for today. First is Dr. Fabio Andres, Application Scientist, Creoptix AG. And our second presenter is Dr. Michael Goldflam, Director of Peptide Discovery with Pepscan. Welcome, Dr. Andres. The presenter ball is yours.
Thank you very much, Elizabeth, for the nice introduction, and welcome everyone to today's webinar. My name, as Elizabeth pointed out, is Fabio Andres, and I am an application scientist at Creoptix Sensors. I'm very excited to hold this webinar today together with Michael Goldflam, with whom we had the pleasure to collaborate for a while now, looking into how GCI, so grating coupled interferometry, the technology that Creoptix developed, can help and make an impact in the field of peptide discovery. So I'd like to start actually telling you a little bit in the first part about GCI. So grating, what is grating coupled interferometry about? GCI as a technology for a broad range of molecular interaction analysis. I will then, after that first part, hand over to Michael, where he would jump in and introduce to you and tell you a little bit about his side of a project about lead discovery and optimization of so-called CLIPS-constrained peptides. I will then, at some point, take the ball back. He will roll it back into my court, and I will tell you a bit more about our collaboration and how what actually we did on our side using GCI, putting it to work, assessing where our technology can actually help in that field of peptide discovery and characterization. So just very few words about who is Creoptix. Creoptix is a growing company, and we are based in the greater Zurich area in a little town called Waedenswil in Switzerland. We're committed, or Creoptix since the beginning is committed to I'd say drive innovation in doing molecular interaction analysis, and to enable some of you guys out there, life scientists, to really accelerate discovery and development by providing tools and technology, cutting edge tools for doing interaction analysis. So very simply put, and this is very simple, simplified schematics, and you might be familiar with that, but we are talking about real-time interaction analysis. What we are actually developing and are offering is a flow-based and a biosensor surface-based interaction analysis technology, where we are working with a sensor surface that can have several different physical properties, where we would immobilize one of the binding partners, here depicted as little Ys, little antibodies, to the biosensor, and then we would flow liquids over it. First of all, of course, there's some buffer phase. We would measure or establish some sort of baseline, and then at some point you would inject what we call analytes, so the other binding partner depicted here as little green hexagons at different concentrations, and we would follow in real time the response that these binding events would produce. We will or will not eventually reach some equilibrium, some dynamic equilibrium, and at some point we will switch back to buffer and follow in real time the dissociation, just to set the stage very simply. And you might be familiar with this kind of interaction analysis from other technologies also. Affinity, kinetic. So what about those terms? Very simply, if we are talking about developing drugs, developing therapeutics, developing diagnostic products, we talk a lot about affinity, and we also talk about kinetics. Often people are very interested in how tight is my compound XY binding to my target. And in a typical drug development pipeline, or also pipeline developing diagnostic products, you would start somewhere defining some hits with a screening method. And looking here at a plot we call rate map or ISO affinity plot, where actually the association rate will be on the y-axis and the dissociation rate on the x-axis, you see that over the course of a classical drug development, discovery and development project, you would go from hits to leads to drugs. And basically what biophysical characteristics would change in that course is mainly the dissociation rate of these compounds, leading to tighter binding compounds that have a higher affinity. If you're able in an early stage in your discovery and your early development project to already look at kinetics, at things like an on rate or an off rate, so really kinetic parameters, you're able to very efficiently from the beginning already also rule out and sort out binders that you would, for example, consider non-specific, having very slow on rates in a combination with a certain off rate. They can still look good in equilibrium methods and show a nice KD. However, if you have the kinetic information, you might have the chance to sort them out very early. So the Creoptix WAVEsystem. The WAVEsystem is actually our main product, and the Creoptix WAVEdelta is that little machine you see here, and it's built on 3 pillars. And I will mainly talk and tackle in the next few minutes on the first 2 pillars. First of all, I will talk a bit about sensitivity and time resolution. I will talk about sample robustness. And if you have more questions about also on the software and the analysis side, we just recently launched a major update of our software that contains quite powerful features for automated data evaluation. We're very happy to also answer your questions and discuss with you, and just visit us. So first of all, sensitivity. What I'd like to show you quickly as, again, very simply depicted graphical comparison is SPR as compared to waveguide interferometry. On the left side, you see depicted an SPR setup. You have a surface that is typically made of gold or silver. You're pointing with a light source, a laser, at that surface from below. And what you create is this name-giving localized field, these surface plasmons. These surface plasmons are a temporary localized field that are actually able to sense changes in refractive index. So when analytes bind to our ligand, and the ligand again is an antibody here, the analytes are the green dots, and that would change the refractive index, and it's sensed by the field. On the right-hand side, you see the concept, and waveguide interferometry is slightly different. We also have a surface, a solid surface. It's a surface-based method. However, now we're coupling the light in to a solid structure we call a waveguide. You can consider that as a sort of an optical fiber. That light runs now below the sensor surface, and what it creates is a continuously running electromagnetic field, and that it's an evanescent field, so it decays to the top, and it covers and runs through the whole sample and the whole sensor surface. And that way, what this setup is able to do is picking up more of the events, more of the analytes binding, and that creates a high signal and quite a high sensitivity. So what we can do with that, just to give you one very simple example, here is you can measure large differences in molecular weight. You can measure, for example, large ligands and small molecules. There's not really a fixed border of how small your molecule can be. It's always a function of how many epitopes will you be able to immobilize on the surface and how active your protein is. What we're showing here is actually more of a showcasing that, of course, when we immobilize a high level, a high density of the carbonic anhydrase, in this case, the protein onto and the surface as a ligand, then we get a high Rmax, and we are able to fit that, of course. We get a high response from that. What we used here actually in the end, however, is a one-to-one model that also includes mass transport limitation. So we get a KT, we get kind of the diffusion constant from the system, because here we are looking at quite fast on rates, and at higher densities, the system is more prone to mass transport limitation. One way of reducing and avoiding mass transport limitation effects is by going lower in your ligand density. That's what we've done on the right side. So the middle and the right plot actually is the very same data set, just the axis is being blown up. And you can appreciate that at very low responses here, below 0.5 picograms per square millimeter, we can still robustly measure those triplicate measurements. So each concentration is actually injected as a triplicate here, and we can robustly fit that to a pure one-to-one model without mass transport limitation. What about time resolution? I quickly mentioned time resolution before. And for that, I'd like to show you quickly on the right side how actually the chip, so that our system is based also on disposable chips that you insert into the machine. And those chips, if you look from top, contain the what we consider as microfluidics, the 4 flow channels with the 4 sensors that you can inject your samples in parallel into. Everything that is considered microfluidics is contained actually on that disposable chip, and that makes the system very robust. So it's basically clock free, so it's a no clocks system. I will speak a bit more about crude sample robustness later, but that's one of the main reasons that everything you consider as microfluidics is on the disposable chip. So it's self-contained and disposable. The system doesn't contain any microvalves that can clog. And by an innovative sample preparation process, we can achieve very fast transitions between 2 different solutions. And that way we get a very high temporal resolution where we're able to measure very quick events, very quick rates. I'm also giving you an example here. With a partner together with the Mario Negri Institute in Italy, we were able to immobilize amyloid fibrils that they were working on and measure kinetics of a small molecule called THT binding to these fibrils. And apparently that interaction has very fast kinetics. As you can see from the left-hand side, looking at the whole data, the whole curves, it looks like a square function. But if you zoom into these regions, however, you can see that we can nicely and robustly fit those quite quick rates with a one-to-one model. And we measure here robustly and with confidence a KD that is almost 11 per second, which is something you could also measure in such kind of a surface-based biosensor can only measure on a WAVEdelta. Measuring off rates, being able to also handle crude samples enables you to do, for example, also something like this, an off-rate screening of crude reaction mixtures. This is also something we will come back a bit later in that webinar when we talk about the collaboration with Pepscan. What we did here, again, with a partner, with one of our customers, we screened actually a small library of around 80 crude non-purified compounds against PDK1. We have a selectivity control here, which is HSP90, just on a parallel different channel to measure kind of off-targeting binding. And we inject an estimated concentration. So these are crude mixtures, an estimated concentration of 20 micromolar. What we could see here is that we can nicely, robustly fit the off rates from those single injections and analyze that in parallel for both targets here. And that could be nicely confirmed also in two publications, 2 peer-reviewed publications. We see an excellent resolution here for the compounds with KD, in some cases well above 1 per second, so really quick rates also we can pick from a crude screening. Of course, we can also do a classical screening approach. We can also, on the WAVEdelta, as you might be familiar from other technologies, we can also look at responses. So that would be an example here for an assay and a project where we looked at responses of fragments. So we looked at a library here together with Rush, which consists of around 530 fragments. And they were screened again against a 25 kilodalton target protein. And what you see on the left plot here is actually the normalized responses. So that plot comes directly out of wave control software, enabling you to normalize your responses, to exclude samples according to the chi-square, for example. And what we found here in this project together with a partner is that also by repeating the whole screening twice, we get a quite nice correlation, and we can nicely reproduce the whole screening. For these 530 fragments, 31 fragments, we had about 2 to 2.5 days of machine time, approximately. So I mentioned also crude sample robustness. So I want to quickly tell you about what kind of samples a Creoptix wave actually can handle. And there's basically not really limits to that, as the system is designed in a way that the microfluidics are actually only on that disposable chip. You never would run into the issue of clogging the system. So the system per se does not have any internal additional microfluidics or an internal flow cell. And therefore, the system can handle really crude matrices. As, for example, serum, plasma, cell supernatant. It stands many organic solvents, DMSO, of course, acetonitrile, just to name a few examples. You can inject cell membrane preps, partially solubilized, unpurified material, particles like virus-like particles, liposomes, nanodiscs, and so on. And of course, you can work with large binding partners with antibodies, nanobodies, SciBodies, nanoparticles, crude membrane preps, etc., you name it. And you have really a reduced downtime if you happen to kind of contaminate the system with very crude matrices. You go on with a desorbing procedure that takes you 15 minutes, and you're up to go for your next experiment. So that's another example, or that's an example actually showing that the wave can actually deal with these crude samples, where together with a diagnostic partner, a diagnostic company, Proteomedics, here we were able to look at a few of their candidates and of their to be developed antibodies. And of course, measuring on the WAVEdelta doesn't give you only the yes/no answer, but it gives you full kinetic characterization already in the crude matrix, which is 90% serum here. And the 10% buffer in the mixture is more for practical reason to compensate for any added volumes of the antibody stock solution we added there. The system actually could also handle undiluted serum or plasma. And so here I would like to hand over to Mike, who will tell you a bit more about our collaboration and from his side of the work. And he will tell you a bit about lead discovery and optimization of CLIPS-constrained peptides. So Mike, the stage is all yours.
Thanks a lot for the handover, Fabio. So in the next 20 minutes, I'd like to give you an overview about the lead discovery and optimization platform for CLIPS-constrained peptides that we built at Pepscan. So Pepscan is a biotech company located in Lelystad. That's 1 hour away from Amsterdam in the Netherlands. And we're working since 25 years in the peptide field, and we offer a broad range of services, including custom synthesis, epitope mapping, personalized medicine, and peptide discovery for therapeutic or diagnostic purposes. And that's what I will talk about today. So Pepscan's long involvement in the peptide field helped us to realize very early that structure is crucial for the function of peptides. And this then led to the development of the CLIPS technology, which stands for chemical linking of peptides on scaffolds. CLIPS allows us to cross-link peptides via the reactions of cysteines with aromatic scaffolds to form monocyclic, bicyclic, or multicyclic constrained peptides. The chemistry to do this is on one side highly reactive, which results in ultra-fast reaction kinetics and very high yields, while being on the other side very specific and requiring only mild reaction conditions, so that no side chain protection is required, which makes this chemistry compatible with biological systems. This is one of the core technologies that I will discuss with you today. The second technology is phage display, and I assume that most of you are familiar with this technology. That's why I will just mention that this approach is based on phages, which is a type of virus that infects bacterial cells and which allows identification of peptide binder for a given target by mimicking the process of evolution. And phage display is a very powerful technology since it allows to screen libraries of billions of different peptides in less than 4 weeks, which was recognized with a Nobel Prize for Chemistry in 2018. It's also important to realize that the ability to screen such large libraries does not automatically guarantee the identification of a good lead peptide for your protein of interest. And the reason for that is that you also have to look in the right chemical space. And this brings us to the question, what is the right chemical space? And to answer this question, the peptide community looked for inspiration in nature. Nature is full of examples of very powerful biological modulators. And here are a few examples, ranging from the human peptide hormone vasopressin, which acts in kidney regulation, to the peptide venom eptifibatide, which is used to manage acute coronary syndromes, and to vancomycin and cyclosporin, both produced by fungi, which are used as antibiotic or immunosuppressant. These peptides cover a broad range of origins and therapeutic indications, but what most of these peptides from nature have in common is that they are macrocyclic peptides and that they incorporate non-natural amino acids. And we understand nowadays that this makes a lot of sense because the macrocyclization decreases the entropic penalty upon binding, while the inclusion of non-natural amino acid modification expands the chemical space that we explore and also increases the proteolytic resistance of these peptides versus proteases. So at Pepscan, we are focusing at a platform that is based on CLIPS phage display, where we discover constrained macrocyclic peptides, and then we are using our synthetic peptide capabilities to improve these peptides further by using non-natural amino acid modifications. And these insights from nature, together with an evolution of peptide display technologies, led now to a growing body of evidence, both in academia and industry, that peptides or constrained peptides have the potential to combine the advantages of both biologics and small molecule drugs in one combined class. This might be a very controversial slide for you if you're not in the peptide field. But due to time reasons, I only want to highlight one example, which is peptide drug conjugates, because these peptide drug conjugates are able to bind with antibody-like specificity to the target receptor, which leads to low off-target toxicity. But due to their small size compared to biologics, they're able to penetrate very fast and deep into tumors, which makes them at the moment a very interesting alternative to antibody drug conjugates. So in the next couple of slides, I'd like to introduce the CLIPS peptide discovery platform that we developed. This platform consists of 4 different elements. The first element is our phage display system that is optimized for CLIPS-constrained peptides, followed by our large portfolio of CLIPS scaffolds, our phage libraries, and finally our platform for peptide validation and optimization. So as I mentioned, the first element is our CLIPS phage display system that is optimized for CLIPS. And it's not a trivial task to combine the required chemistry and biology. And that is why, because some phage coat proteins, for example, contain cysteines, and these cysteines can interfere with the chemistry or the chemistry with the biology. So to address this, we developed a model system that is based on these 2 phages depicted in green and orange on the left side. And each of these phages is presenting one single type of peptide at its tip. And this peptide is able to bind with high affinity and specificity to the target protein depicted in the same color. But only if the peptide that is present on the phage is cross-linked successfully with the right CLIPS molecule. This model system allows us then to mix the phages together in ratios that mimic the conditions in a phage display library scenario, where we have a tiny fraction of binder that are diluted in a huge excess of non-binder. And then we can do a target pull-down experiment that we can quantify by qPCR. This assay allowed us both to benchmark, but also to optimize our platform up to the level where for some targets we can find CLIPS peptides in a single round of selection from our billion member libraries. I don't have more time to go into this in detail, but please contact us if you like to have more information about this. This phage display system comes in the context of our large portfolio of CLIPS scaffolds, and these CLIPS scaffolds allow us to cross-link the peptides into macrocycles during phage display. We focus on monocyclic peptides, which allows us to use symmetric CLIPS molecules, which ensures that we only get one single peptide product. But it also has the advantage that there's a large structural variety of the symmetric CLIPS scaffolds, which translates to a large structural variety of peptides that we create, because these different structured CLIPS molecules impose different geometrical constraints on the peptide and also foster different type of interactions with the peptide side chains. And generating structure is important because the structure of a peptide determines to a large degree its binding behavior. So having the ability to explore a larger structural space increases the chance of identifying more potent binder and the targeting of more diverse epitopes. So the next element in our library is our phage library. Our first library design is based on 10 randomized positions that are flanked by 2 cysteines to give monocyclic peptides. We use a codon-balanced approach to ensure an equal distribution of all amino acids on all positions, excluding cysteine and stop codons. So it's a high-quality library. And this library has a diversity that exceeds 10 billion and was validated by NGS sequencing, as you can see on the left side. We do have now additional library formats available, and these include now the possibility to conjugate a molecule to the N-terminus of the peptide during phage display. This could be, for example, a lipid, a carbohydrate, or molecule from a fragment-based drug discovery campaign. This platform can be used in, to serve a broad range of applications. On the therapeutic side, we can discover peptides for targeting purposes, like peptide drug conjugates, or for the fusion or conjugation to biologics. So these biologics gain a function, or to add, for example, a cell type specificity. But we can also discover peptides for the direct use as therapeutics, targeting, for example, protein-protein interfaces, enzymes, or GPCRs. On the non-therapeutic side, our platform can also be used to develop diagnostics or reagents for purification technology applications like antibody purification or the removal of antigens from food streams. So here's an example for a lead development plan for such an application. It starts by understanding the exact project goals in terms of affinity and specificity, but also if, for example, cross-species reactivity is required for proof of concept studies in an animal model, the targeting of a specific epitope, or, for example, if a pH-sensitive binding mode is required. Then we use this information to design a selection strategy that is then realized with our CLIPS phage display platform, running multiple library scaffold combinations in parallel. The phage display endpoints are then analyzed by NGS, and this data is then used to generate lists of 10,000 candidates. Then we start to pick a couple of hundred of candidates from this list, which are then synthesized as crude peptides and retested for target-specific binding in ELISA type assays. This allows us then to narrow the funnel further down to a set of example 20 peptides that are then resynthesized and re-evaluated in a purified format for binding, for function, but also, for example, proteolytic stability. And this gives us then our first set of lead peptides. And these lead peptides can then be further optimized either on peptide arrays or as crude peptides, also by inclusion of non-natural amino acids until the peptides match the requirements of the project. And once that is the case, we can then provide our clients large quantities of these peptides for pilot experiments or clinical trials. During this process, we rely mainly on ELISA, but I think it is important to corroborate the results, at least during the lead identification and the lead optimization stage with an alternative technology like SBR or GCI. So In the next couple of slides, I have an example from such a collaboration that we had with Abreos Bioscience, a precision medicine company from the States. So Abreos has a pipeline that is based on peptides that are able to bind with high affinity and specificity to a biological drug in a drop of patient blood. And this allows them to quantify the concentration of that drug. And it's important to ensure that the concentration of the drug is inside the therapeutic window. To low concentrations can diminish the therapeutic effect, while to high concentrations can lead to toxic side effects. And patients catabolize these drugs at different rates, and the ability to fine-tune the dosing on a patient-specific level increases both the safety and the efficacy and reduces the cost during the clinical trials and during the treatment. And Abreos has a very ambitious pipeline where they want to produce this type of assays for all the major biological blockbuster. And for this, they have their own phage display platform that is based on linear peptides or disulfide-constrained peptides. And for many of these blockbusters, they were able to find peptides that serve this purpose, but some of these targets turn out to be too challenging for their platform. That's where we then joined for a collaboration with Abreos, where we use our CLIPS phage display platform for these challenging cases. So for this project, we selected the 6 scaffolds that you can see on the left side. We combined it with the same monocyclic phage library, and then we performed 3 rounds of phage display selection using an FC depletion strategy to ensure that we only find peptides that bind specifically to the biological drug, not any other serum proteins. After phage display, we followed up by NGS and by bioinformatics. So during the first 2 rounds of the selection, we only observed low titer of recovered phages, but then after round 3, we actually saw a significant increase in recovered phages between 0.5 to 6%, which indicates that an enrichment process did take place. So we decided to sequence round 3. What you can see here is the cumulative abundance of unique sequence reads in the NGS population. And as you can see, the most abundant sequences represent between 25 and 95% of the NGS population, confirming that a strong enrichment did take place. And NGS can handle very diverse populations, so probably less rounds of selections would have been better, but nevertheless, this provided a lot of data to move forward. So we analyzed the outputs, and what you can see now on the right here is a consensus motif of the major peptide sequence cluster. And the first thing I like to draw your attention to is that we observe different peptide cluster based on which library scaffold combination we used, which indicates that the scaffolds indeed led to different peptide conformations. But you can also see that these peptide motifs contain some similar features. So a lot of them contain this double red motif, DE, DD, or EE, that is often flanked by these 2 black amino acids, FW or VF. This probably indicates that some of the peptides share an overlapping binding site on the target, which does make sense because since we're using an FC depletion strategy, we expect that the majority of these peptides do bind to the CDR region of the antibody. And if we could only pick 6 candidates to move forward, then this would be my choice. But then we would neglect all the wealth of information that we generated by NGS. Therefore, we decided to select 450 peptides for each library scaffold combination. These peptides were synthesized on peptide arrays, and they're probed for binding to the target and isotype control. What you can see here in the middle is a single line for each of these 2,700 peptides, and the length of the line corresponds to the fold binding for the target versus the control. So the longer the line, the better. And as you can see here, this target protein seemed to have a preference for the first 3 CLIPS scaffolds and especially the last CLIPS scaffold. Based on this data, we started then to cherry pick candidates. And very early then we found peptide 2 here that you can see on the right that was retested as purified candidate by ELISA. And we could confirm that this peptide is binding with an EC50 value of 35 nanogram per milliliter and complete specificity to the target drug. So we decided to focus on this candidate, and thanks to the nice collaboration we had with Creoptix, we were able to confirm with GCI that the peptide is binding indeed in a very clean 1-to-1 binding mode to the target protein and with an KD of 670 nanomolar. We're also able to confirm that the peptide is binding still in the presence of serum. It's still a specific binding event, but the KD dropped slightly down to around 3 micromolar. And these measurements were actually done in duplicate, and I do remember that I was quite surprised to see that the reproducibility of the data is so nice. It's actually very difficult to see 2 distinguished lines. And also I found this measurement in serum quite interesting because obviously all therapeutic and diagnostic applications are performed in the presence of body fluids. I'm curious to see if this approach helps to translate between in vitro to in vivo results in a more meaningful way. In terms of the project, a KD of 670 nanomolar might seem not to be very impressive. For other confidential projects, we usually obtain ligands in the single or double-digit nanomolar range. But as I mentioned, Abreu's already tried to find peptides against this target with the on-phage display platform. So I think it's fair to say that this is a challenging target. Further, in the GCI setup, we did not make use of the avidity of the biological drug, but that's actually the case in the ELISA and also in the diagnostic setup. And Creoptix told us, Abreu's told us that they were actually able to use that peptides to detect the drug in the presence of serum in a concentration range that is covering the therapeutic window. Therefore, I think it's actually a very promising lead peptide. We wanted to understand the nature of that peptide further, so we retested that peptide as a linear variant and as an SS oxidized variant, with the result that none of these variants were able to bind to the protein anymore, which makes sense because, as I just mentioned, Abreu's already tried to find linear SS-constrained peptides that were binding to that protein, and they couldn't find any candidates, which confirms that by using this CLIPS molecule, we're actually looking in a different part of the chemical space. Further, we tested a couple of variants where we replaced now the CLIPS scaffold with a set of alternative CLIPS molecules, and these variants were then retested for binding, with the result that the majority of substitutions either abolished or greatly reduced binding, highlighting that the nature of this CLIPS molecule is very relevant for the binding of this peptide. And then further, we tested all possible bicyclic variants of these peptides with the result that none of them showed binding anymore to the target, which implies that this peptide or the similar peptides would not have been identified from a bicyclic peptide library, but of course this does not exclude the possibility that there could have been other bicyclic peptides that could bind to the target. So in case we have to optimize peptides further for an application, then we can either use our peptide area platform, crude peptide library, or crude peptide libraries for this purpose. And this platform is suitable for many different peptide formats, including monocycles, bicycles, or for even more complex geometries. And we usually start the lead optimization process with a full replacement scan. And this means that we take the lead sequence that you can see on the left here, and then we generate all possible single point mutation, excluding cysteine replacements. And these variants are then synthesized on the arrays and probed for target binding. Then we often can see 3 different types of replacement behavior. So for the positions that you see in green, any replacement completely abolishes binding, highlighting that these positions are part of the core binding motif. Then we have the positions in white, where any replacement does not really affect the binding of these peptides, meaning these are neutral positions. But then finally, we also find positions that are highlighted here in orange, where actually we find substitutions that increase the binding of the peptide. Then we can take this information about these improvements to design an optimized version of that peptide. And by doing this, we were able to find to improve the affinity of this peptide by a factor of 7, which is only a moderate gain in affinity. But after this, we can repeat this exercise using non-natural amino acids And here we're able to find two non-naturals that were replacements for the core binding motif. And by using these two modifications, we're able to improve the affinity down to 310 picomolar, which represents a 190-fold improvement compared to the original lead peptide. And I, as a chemist, find it quite striking that we moved only one methyl group from isoleucine to norleucine, and we added one methyl group from alanine to the aminobutyric acid to generate so much more binding energy. So what we see quite often in our optimization campaigns is that when we look in the natural amino acid space, we only get small to medium gains in optimization. But then when we move to the non-natural amino acid space, we can get very large gains in optimization. And this makes sense because a big part of the natural amino acid space was already explored during phage display, but not the non-natural amino acid space. Further, the inclusion of the non-natural amino acids also increases the proteolytic stability of the peptides. This data helps to understand the binding mode of the peptide, which is not only useful for optimization, but also, for example, to file IP protection on your lead peptides. And with this, I come to my summary slide. So I explained the CLIPS technology, which is a very powerful chemistry to generate redox-stable macrocyclic peptides. And because it's a very selective and clean chemistry, it simplifies the whole process of discovering, optimization, and manufacturing of these peptides. We can identify peptides for a given target protein using our phage display platform that was fully optimized for CLIPS peptides. And we can do that both using traditional multiple selection round approaches or also single round approaches. And this comes in the context of our large portfolio of CLIPS scaffolds, which allows us to vastly expand the chemical space that we're exploring. And together with our high quality phage libraries that exceed over 10 billion variants. Then we use NGS analysts to really dive deep into the data that we generate. And based on this data, then we can select a lot of candidates that we can validate and optimize on our platform with inclusion of non-natural amino acids. And we can cover this complete peptide discovery pipeline via a single company up to the GM production of these peptides. And with that, I would like to go back to Fabio to give you some more insights on our collaboration and how we can use the GCI platform to validate peptides.
Thank you so much, Michael, for the interesting insights onto your side. I will, as you mentioned, that here, I guess, yes. So for one of the projects coming or being carried out at Pepscan, where they did lead identification or hit identification with ELISA, around 200 CLIPS peptides were screened by ELISA, as Michael has just shown you how the platform works. And in their assay, actually, the setup, that's something you might want to keep in mind, is that the peptides are biotinylated and are the immobilized partner, which are coated or immobilized on the ELISA plate, whereas the bivalent antibody is binding over it and is the detected binding partner in the ELISA. So for one of the projects, actually, Michael identified and picked out 6 interesting hits that were coming out of the ELISA, so showing positive binding of ELISA, and he picked different sequence clusters and different scaffolds, so 6 interesting hits that were then transferred to us, and we characterize them on GCI doing full kinetics. This is what I want to show you on the following 2 slides. So looking at the first 3 of those 6 picked out hits here, we see that GCI can confirm that these hits are actually really binding. Also on our technology, we see the real-time binding and can characterize the full kinetics here, of course. The other 3 are on the next slide, but I can already tell you that 5 out of 6 of these picked hits could really be confirmed on GCI here. What we observed is quite a high diversity of kinetics. the peptides overall had a low micromolar. We saw a low micromolar affinity here. I will come back to that and comment on that a bit later. But we saw quite a diversity, which was interesting in kinetics in the rates themselves. Try to activate my pointer very quickly here. Exactly. And then what you can also appreciate here is that what we see so far, and that's still under investigation. So I give you here a glimpse into a running project, into an ongoing work. What we see so far is that some of the peptides still show some kind of heterogeneous behavior, so they're not nicely fittable with a pure one-to-one model, and you get a bit of better fitting with a model that has a bit more degrees of freedom. And I don't want to do model hopping here, but it's just to mention that we see some slight heterogeneity here in the kinetics, and that could be, for example, it's just speculation, could be related to the purity level, side products, or non-specific binding at higher concentrations. Looking at the 3 more of those ELISA hits, we see also ELISA hit 4 and 5 could be confirmed. They show nice dose response and dose respondent binding, and they also show curvature, so we can really zoom in and see that's actually really real-time binding we're observing there. Where a 6th hit, you can see gives some kind of signals on our device. However, there, if you zoom in, you don't see any curvature, so there we would have a low confidence and we would consider this a potential false positive here. So one simple question to ask actually, would be, how good is ELISA at detecting true binders? So one notion and one observation we made is that we do not see really a good correlation for the ELISA EC50s and the KDs measured on GCI. So the sampling number here is really small, so I do not want to stress the term correlation too much, but as you can see, EC50s and GCI KDs do not really go well together. There, we have to be aware that the setup is slightly different. This is also under investigation. We might also turn the assay around and see if we get better correlation by other assay setups. On the right-hand side, what you can see is that, and I'm not showing you that additional data here, that we also tested the crude peptides, so the crude reaction mixtures of those 5 confirmed ELISA binders. There, of course, you cannot measure full kinetics, as you cannot do the titration of the crude full reaction mixtures, but you can do that one injection and fit the off rate. And I will show you a bit later how we can use that also for screening approaches. And what we see is that actually the off rates determined from the crude peptides compared to the off rates we got from the full kinetics on the purified peptides, they go quite well together, and we see a nice correlation there within that small data set, of course. So, but that kind of motivated us really to go a step further. I like to say, as the WAVEdelta, as I told you in the beginning, can handle crude mixtures and crude reaction mixtures, as you saw on the previous slide also. And we decided to go for a full grating coupled interferometry screening on the wave of the CLIPS peptides, around 200 that were screened in ELISA, and really do the full screening here. Again, the target here, the antibody, a bivalent protein target is immobilized on the surface, and the crude peptides are sequentially injected over it. What we can do, and what I want to show you in the next slides, is that we can do a response and an off-rate analysis, and probably even combine those evaluation strategies. First, I want to mention a few challenges that you can encounter, especially when you work with peptides. And it seems like here some, or at least one of the scaffolds we're using here for these CLIPS peptides seems to have quite a high positive net charge. And what you see here is actually a non-specific binding onto the sensor. So the peptides on that sensor we used here on that surface, where we did amine coupling, of the target protein has a strong negative net charge, and so strongly positively charged scaffolds would give you non-specific binding. In blue, you see that's kind of the measurement channel with the target, and in yellow, you would see the reference channel. That means that the positively charged scaffolds bind non-specifically here. So there's a few options, there's a few strategies that one could try and that we're working on also further. You could use a Switter-ionic surface. We offer a Switter-ionic surface we are calling PC-set wave chips we have in our portfolio that have a more balanced charge distribution to look at these kind of scaffolds. Currently, what I'm showing you here, these guys are excluded from the analysis. Another challenge that you could encounter actually is tight binders. So if you get in a system where you do not have regeneration conditions as here, if you get very slowly dissociating peptides, of course, what they would do is they would saturate the binding sites and affect then the following peptides in their that lie actually in their dissociation. So here there's no regeneration conditions available, and so what we did is some kind of a library cleaning. So we rearranged the injection order and sorted those candidates to the end here. So what you can do then, of course, first of all, you can look at responses. You can look at what responses these peptides give on the target. What you see here is on the left-hand side, kind of the not normalized, more like a raw signal. The responses are plotted on the y-axis here, and on the x-axis, you see all the different peptides. You see a first plate, a second plate. What you can also appreciate here is that the target activity as measured by a positive control, and this positive control, I have to mention, is a purified peptide that we characterized before, which serves as a positive control. And you can see that on the second plate, actually, the target activity starts to drop. So also here, for the further analysis here, we made a cut and analyzed the screen up to this point. Where here we use the threshold of a normalized response of above 0.2. And of course, on the other hand, you can look at off rates, as I showed you before, and I will show you also on the next slide. You can inject the crude reaction mixtures and just fit the single off rates and get an idea already from a screening about the kinetics. Again, charged scaffolds were excluded here. What we see is that also here the control, the positive control peptide behaves very reproducible up to a certain point. So mainly on the first plate and up to this point, it shows a very, very high reproducibility in terms of the off rate that we find over the screen. That kind of validates the data, or at least part of the data here, that we then used for further analysis. So here you can see actually from that off-rate screen just a few picked out examples and how that looks, and you can see that we get good fits. So the black lines are actually the fits over those response curves, and we get good fits across the whole range of detected KDs. So, and what we did is an approach then of combining kind of the response and the off-rate analysis with the goal to come up with the most interesting candidates for further analysis. And so the approach here is that we plot on this plot, we're plotting on the y-axis the response level that these peptides are giving, and on the x-axis we're plotting the logarithmized off rates. And we decided to kind of, or we tried to dissect the plot into several regions and did 4 quadrants, whereas on the first one, top left, you would see candidates with a slow off rate, giving a high response, giving you a high confidence that this would be good binders. Bottom left would show slow off rates, but a low response. So those could be good binders, but we have a lower confidence. Quadrant 3, top right, is rather fast off, but a high response. So there you have a higher confidence that these are actually true binders, but weak binders. And in the 4th quadrant, you would find probably weak or no binders with quite a high confidence. So what we did here so far, and I mentioned it again, that this is an ongoing project still. What we did so far is we selected a few hits from quadrant 1 to 3, and we purified them. So on Pepscan side, so Mike actually purified them, sent them back to us for further characterization. And what you see in the following here is actually that panel of hits from that were identified by GCI that we selected and purified. And you can see that we are able to confidently say about for half of these peptides that they can be really confirmed as true binders, whereas 6 out of 13 peptides behave a bit less clear, I would say. Some show a bit of promiscuous behavior, show a non-specific behavior, or only bind with a high signal at very high concentrations and do not really scale with the concentration. So the peptides in this box we would consider either promiscuous or also put in the bucket of false positives. Also here, you can appreciate and needs to be mentioned that some of them actually show a bit of heterogeneous behavior and are not fittable very cleanly with a one-to-one model. And this is an observation to be investigated now, of course, and it could be related, for example, to the purity level or some sort of non-specific binding of the peptides. So this will bring me to my summary and conclusions so far for that collaboration. The majority of selected and purified ELISA hits could be confirmed on GCI. That's what I showed you in the beginning. We could identify further candidates, further hits by crude peptide screening using GCI technology on the WAVEdelta. The characterized GCI hits were not found by ELISA, so this needs to be stated. So there seems to be a difference, and we seem to find, seem to find a complementary set of binders. However, both methods, both technologies are able to identify hits and lead candidates. So one key difference between the assays, of course, we have to keep in mind is that the ELISA has the peptides on the surface and the target flowing over, so it would be also prone to avidity, for example. Generally, what we saw is a bit of lower affinity of the hits found by ELISA when we measure them on GCI versus the EC50s measured by ELISA itself, and that's actually that would actually make sense because of the avidity you see in ELISA and we would not see on the GCI in that setup. So further assay optimization is surely possible here. That's work in progress, as I showed you. And I think it's fair to state that GCI on the WAVEdelta can be used for hit validation. So we could validate hits that came out of the pipeline and out of the screening from ELISA at Pepscan. And as well, GCI seems to be a complementary discovery technology for those kind of peptides. And with that, I would like to quickly mention that on Creoptix.com, you actually are also able to get much information about our company products. You would find application notes, publication lists, and also get news and upcoming events, of course. Visit us on Creoptix.com. You can also visit us on Twitter or on LinkedIn, if you like. And of course, as seen on the last slide that we're actually sharing, if you have any further questions, we're very happy to answer them. If these questions come up a bit later, please do not hesitate to contact us directly or me directly, and I will be happy to answer any kinds of questions. Thank you.
Thank you very much, Dr. Andres and Dr. Goldflam. We have run over, but I'd like to take just a minute or two to ask just a couple questions. Our first question, can you elaborate on how to choose an appropriate immobilization or capture strategy for protein binding?
Yes, thank you very much for that question. So there's a broad range and a variety of capturing and immobilization techniques that you can use. And I think, yeah, one of the main parameters to look at is, do you have any tags? So if you have any tags fused to your proteins, of course, you can use them. For example, a his tag can be captured on a nickel NTA surface, or an FC tag can be directly captured on a protein AG wave chip, for example. So the tags actually enable you to capture your proteins under also very mild conditions. On the other hand, of course, you're also free to use any classical coupling strategies that would lead to covalent coupling onto the sensor, such as amine coupling. So in the end, this is really something that depends on what tags you have available, and to decide for the optimal strategy there.
All right, thank you. And just one more question. I'll plan on sending the rest of them along to you via email. What is the best CLIP scaffold for therapeutic peptides?
Okay, thanks for that question, Elizabeth. I think that's always target dependent. And if you don't have any further information on the target, you just try to pick CLIP scaffolds that give very different geometric constraints to increase our chances to find a good peptide, a good lead peptide. If we have some information on the target that it's, for example, could have a binding site that could bind to, I don't know, an aryl or naftual moiety, then we would choose one of these scaffolds.
All right, thank you so much. I do apologize that we don't have any time for any further questions, but I will be forwarding them along to our presenters for answer via email. I'd like to thank our two presenters today, Dr. Fabio Andres and Dr. Michael Goldflam. I'd like to thank our sponsor for today's webinar, Creoptix AG. And most of all, I'd like to thank those of you who came and spent this time with us. We really appreciate you being with us. We know times are crazy and you're being pulled in many different directions, so we really appreciate you coming and spending this time with us. So on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, I'd like to thank you all so very much and wish you a wonderful rest of your day. Thank you all.